my name is Tanya Buxton. I'm a biologist. I live in Northern California, and welcome to my lab. I've been working in the field for about 25 years. I'm really excited about research in biology and use of new technologies. I believe that anybody can be a biologist. I'm happy to have you here today in my lab, and I hope that you'll find these videos useful and that you can do some of these experiments on your own. Today we're going to talk about CRISPR, which is a new technology in gene editing. It's been all over the news, you may have seen, and it really has great potential. CRISPR stands for clustered, regular, interspersed, short, palindromic repeats. It's an acronym that is being used for explaining about a new discovery in gene editing that's done in bacteria. But really, there's an enzyme called Cas9 in the bacteria cell that's actually doing the editing. So what really is CRISPR? It is a gene editing system. And here I have a piece of DNA that has a break in it. DNA gets damaged in a cell, and the cell has mechanisms in place to be able to fix that piece. It's a break or a cut, and existing material within the cell is used to replace that piece that's missing. This is a normal, natural process. So in the CRISPR new technology, what is trying to be done is to create a break or a cut at a certain spot and insert a gene or piece of DNA of interest. And we will trick the cell by flooding the cell with many, many pieces of the gene of interest or the piece of DNA that we're wanting to insert into our genome or DNA. And the cell will use its normal mechanisms, but replace that cut with a piece that we're interested in. So we need to give the cell a way to cut at a certain spot. That's the Cas9 enzyme. And we also need to provide the cell with the gene that we want to insert into the genome. I'm going to explain a little bit more about the molecule of DNA and how it works and what it does in a cell. So DNA is a double-stranded helix molecule, and it has chemicals that match up or pair up together. You may have heard of adenine, thymine, cytosine, guanine, referred to sometimes just as A, T, C, and G. They pair up in a double strand. And the job of DNA is to make another chemical called RNA, which is a single helix molecule. And RNA is carrying the code from the DNA. And DNA, we sometimes will say a series of these chemicals can be referred to as a gene. The RNA takes that signal to a ribosome. A ribosome is a kind of part of the cell, and its job is to create proteins off of the map that the RNA brings along. In this particular CRISPR experiment, what we're doing is introducing a piece of DNA that's going to make the bacteria be resistant to an antibiotic called streptomycin. We are actually going to disable just one spot on the DNA, just make a one-point mutation for a gene that normally makes a protein that makes up a ribosome. So when a cell is exposed to streptomycin, an antibiotic, the antibiotic will actually bind to one of this particular RNA ribosome protein, and it will kill the cell because it's not allowing the cell to make its ribosomes, and ribosomes are incredibly important because, as you can see, they're used to make all the proteins that the cell needs to make. So we're going to be disabling just one spot on this gene, and that's going to make streptomycin no longer able to bind to this particular ribosomal protein, and the cell will then be able to create its proteins just like it normally does, and it will be considered streptomycin resistant. Let's walk through the overall steps of the experiment. So first, you're going to need to make some Petri plates with agar in them to allow bacteria to grow. So you'll make the agar chemical up and pour those into plates. Next, you'll need to grow some bacteria on those plates. So we'll take E. coli from a culture, grow them onto those plates. And then the cells, those bacteria cells, need to be made what we call competent, which is allowing them to uptake DNA from the environment. And then once they're competent, we'll use a chemical called calcium chloride for that. And then we will add DNA. And in order to allow the cells to actually take in that DNA from the environment, we need to give them a heat shock. We're just going to change the temperature slightly, just for a few seconds, allowing the DNA to come into those cells. Then we will give the bacteria some LB broth, which is a food that will nourish them. And then we will put them back onto some plates again, some agar petri plates. And finally, for 24 hours, we will put them into an incubator at 30 degrees Celsius and allow them to grow. And then come back and look at our results. So you're going to need some large equipment. Some of the equipment you can get on eBay or a large storage house. And you'll need an autoclave, a microwave, incubator, a balance, a water bath, and those are some of the bigger pieces of equipment that are more expensive. You'll need a Bunsen burner to sterilize things, and a set of micropipettes. They're actually micropipetters because they're used to pipette very small micro unit volumes. I recommend getting 3 different sizes: a P20, a P200, and a P1000 is what they're called, and that shows the range of their volumes that they measure. There's some consumable items that you'll need. Most of this is plasticware, tips for those micropipetters, little micro tubes, petri plates that have already been pre-sterilized. You can buy the spreaders for the bacteria and the inoculation loops for the bacteria as consumable items, or you can get a more permanent version of those. It's always good to have a Sharpie pen to label things. And the reagents that you're going to need are your stock of your E. coli, which comes in a stab, which is just a bit of agar where the culture has been stabbed in using an inoculation loop. You'll need calcium chloride, the salt compound, the LB broth, and a powdered compound. LB agar comes as a powder to begin with as well. And you'll need the antibiotics, streptomycin and kanamycin. Those also come in powder. They are somewhat restricted in terms of buying them. And then you'll need some ethanol, alcohol. That's going to be used for sterilization. And then you'll need your DNA of interest. In this case, we're going to have 3 types of DNA. We're going to have a plasmid, which is the Cas9 plasmid that contains the genes for the Cas9 protein. We'll need an enzyme that has the DNA that has the guide RNA capability. And then we also need our template DNA, which is what we're going to be using in this case, the streptomycin resistance. Let's talk about the safety of doing CRISPR experiments. So some people may think it's quite dangerous, but I want to assure you that what we're doing in this lab is quite safe and not harmful to us or to the environment. It does have potential to be harmful. It does have potential to change human genes or to create some kind of a resistant bacteria or viral strain. But we're going to be working in little tubes and with petri dishes, and we will be making sure we take proper precaution and also dispose of things properly. There's really nothing that would be harmful to us or to the environment with what we're going to be doing. We're going to work with bacteria. We're not working with human cells or animals. And the particular strain of bacteria we're using is an E. coli strain that is not a pathogenic strain. So it's not the scary E. coli that you hear about that can make you very sick. And we'll be manipulating genes within this E. coli, but we'll make sure that we don't let them out into the environment when we're done. The chemicals that we're using are not particularly harmful. We will take a few precautions, but actually we don't even need to worry about protecting our skin or our eyes too much with these chemicals. So I just make sure I wash my hands before and after the experiment. And The only chemicals that we are going to use that are restricted agents are antibiotics. You will need permission to buy those through a chemical storehouse. We will handle those properly because they are antibiotics. They could, if they were to get into our system, kill bacteria within our system. And finally, we're going to make sure we dispose of everything properly at the end. So we'll collect bacteria at the end and put them into biohazard waste bags and have that disposed of properly. One of the pieces of equipment that you're going to need to have to do a crisping experiment is an incubator. And it's a box that's set at a certain temperature, and we'll be setting this at 30 degrees Celsius for this experiment. You're going to grow the bacteria in this chamber. You're also going to want to have a microwave that's dedicated for experiments and lab equipment and not food. You'll be using this to melt agar that you're going to pour into Petri plates, and so it would be a good idea to have hot pads because this will be hot when it comes out of the microwave. This is an autoclave, and I happen to have a Brinkman model, but there are quite a few different brands that you can use. It's for sterilizing your equipment, your reagents, and plasticware. And dentists will often use these, and they have a smaller version, but you want to make sure that you get one that's the right size to fit your bottles and your materials. It'll have a chamber that you will put your materials into, so just make sure that's the right size. There's plenty of different brands though. You're going to want to have a water bath, and what it is a chamber filled with water. There's a rack in there. We'll be setting this at 42 degrees Celsius today. We'll be putting in tubes into that rack, and it will be for a heat shock. We're going to give the bacteria about 30 seconds of a slightly warmer temperature in order for them to take up the DNA. You're going to want to have a digital balance, and it's very good if it has an accuracy of 1/10 of a gram. We'll be measuring out various chemicals to make solutions and media with the scale. There are some less expensive versions that are battery operated, but again, it does need to have some accuracy up to 1/10 of a gram. You'll also want to have a weigh boat because you'll be measuring out the chemical, and a spatula would be good. And when you put the weigh boat on there, you can zero out the scale or tear it, and then start adding your chemical, and you'll know the weight of that chemical. This is a Buxton burner. You can use a traditional one if you have a gas line or a little propane tank type. And you'll need a lighter or a striker. And we'll be using the flame to sterilize things. So we will use what's called an inoculating loop. We sterilize that. We will also use a glass spreader. I need to sterilize that with the flame. There are plastic versions for a spreader and a inoculation loop that you can buy. It's just a little bit more wasteful, and they're single use. Some of the plasticware that you're going to need are some little tubes, some micro tubes, and some tips that go with the micro pipettes. And there's two different sizes, this type here and this type here. You'll also want some containers, some glass bottles with labeling tape. Sharpie's always good. And maybe some containers for your waste and for your sterilization. Some tubing would be good as well. And then for these tips, you need to have micropipettes. So there are 3 different size micropipettes that I recommend. One of them goes from 1 microliter to 10 microliters for its range. The next one is a 10 to 100 microliter range, and finally there's a 100 to 1,000 microliter range. There are various companies that make these micropipettes, and so they may look more like this. These, you actually have to dial in the amount. And for these you have to dial in the amount. For example, this pipette has a range of 20 to 200 microliters, so if I were to set this at 100, I would get a 100 microliter volume. And then you do have to put a tip on the micropipette and drop your liquid in the tip. So the tips are not reusable, and that's why it's helpful to have a waste bin to deposit the tip. There are different sized micropipettes that you can get to be able to dispense very, very small volumes. And in this lab, we're going to need to use very, very small volumes because we're working with DNA and we don't have a lot of it. So this first pipette has a range of 100 to 1,000 microliters, and it has a dial, so you can dial in the amount you want. So I've set it at 200, so it'll be 200 microliters. This one uses the blue tips, and you put on a tip, close the box because generally your box is going to be sterile. Let's use just pipette some water for now. So I have some sterile water here. I'm going to push this plunger down until it has a little bit of a stop, and then I'm going to put it into the liquid and release the plunger, and that will draw up liquid into the tip. And check that you can see liquid and that there's no air bubbles. You may want to dispense that liquid into one of the microfuge tubes. So open up the microfuge tube, keeping it sterile on the inside, and then dispense the liquid by pushing down to a second stop that you'll feel when you push down the plunger. Take that out while you're still holding onto the plunger and close your cap. You can set it down into a little rack if that makes it easier. This tip now can't be reused, but there is a little dispense button that you can press on and shoot off the tip into a waste bin. If that had bacteria on it, you'd want to put it into the biohazard. So that's the first kind of micropipette pipetter. There are other size micropipetters. Here's another pipette that has a different range, and we're going to set this one to 88, which will give us, the range on this pipette is 10 to 100, so 88 microliters. Put a tip onto that. And again, we'll use some distilled water. You want to not set the tip on, touch the tip to anything where you might contaminate it. And go to the first stop with the plunger, draw up your liquid, closing this bottle to keep it sterile. Taking your tube and opening it and dispensing the liquid. I like to be able to see that the tip is positioned onto the side of the tube and watch the liquid go in. Remove the tip so that I don't drop back any liquid, and close my tube, and I can get rid of that tip. The reagents for this particular experiment are to have a bacteria culture. So this is called a stab, where there's a little bit of agar in this vial and bacteria is growing in there. This is E. coli. And then you're going to need to have the media that the bacteria grow in. So you'll need to have LB broth media and also LB agar media for the Petri plates. And LB stands for Luria bertani, which is the name of a broth that's got nutrients in it for the bacteria to grow. You're also going to need some antibiotics because we're going to select the bacteria that actually did get crisped. And so today we're going to be using streptomycin and kanamycin as our antibiotics. Those are controlled substances, so you will need to get them from the proper chemical storehouse. You will also need calcium chloride to make competent cells, and you will need the proper DNA for the experiment that you're doing. So today we have a plasmid that has our Cas9 enzyme gene on it. We also have the target DNA that we're going to be inserting into the cells. And we also have a plasmid that has the capability to make the guide RNA to cut the chromosome DNA where we want it to be cut. So I'm wearing a white lab coat today, not just to look like a scientist, but also to see if I've spilled anything on myself. And we're not using anything particularly harmful in terms of chemicals today, but you do want to be making sure that you're washing the coat afterward and not putting these chemicals out in into the environment. Also, I wash my hands before and after the experiment. It's not necessary to wear gloves with this experiment, or even goggles, but just you want to be cautious and be clean about what you're doing. We don't want to introduce contaminants into our experiment as well. Let's go into a little bit more detail on each step of the procedure. So first off, you're going to need to make some agar and then pour that agar into Petri plates. So we will be making the agar and then as a powder with a liquid, and then sterilizing it in an autoclave, and then we'll sterilely be transferring it into already pre-sterilized plastic dishes called Petri plates. In order for our bacteria to grow on Petri plates, we need to give them food. So we're going to make LB agar, which has a nutrient broth in it, as well as a gel-like substance that gives them moisture so that they can grow. You need to weigh out the LB agar powder, and you'll need to use your scale. So zero out your scale with your weigh boat, and we're going to use 7 grams of we're going to weigh 7 grams of the LB powder, and then we're going to transfer it into a glass bottle. Use the weigh boat as a funnel. And for every 7 grams, we'll add 200 milliliters of water, distilled water. Swirl that a little bit around. It's okay if it doesn't get totally dissolved, because when we put this in the autoclave, that will heat it enough to dissolve it. And you always want to label your bottle appropriately. So I'm going to write on a piece of tape LB agar. And I'm also going to put my initials and today's date so we know who made it and when it was made. And this will need to go into the autoclave to get fully sterilized before we pour the plates. You're going to also need to make your LB broth in the same way that you made the LB agar. So you'll take the broth media, you'll weigh it out on the scale, you'll put it into the bottle, and then add the appropriate amount of distilled water, and then we need to autoclave that, just like we did. So I have here LB agar, the calcium chloride, some water, distilled water, and LB broth. And you put that autoclave tape on the lid and take them over to the autoclave. This is a liquid sterilizing, so we need to fill the chamber with some water. And your autoclave will typically have a fill water line where water from the tank here. And by the way, you want to make sure before you start that this tank has been filled with distilled water. And you're going to watch water come into the chamber, and there'll be a little line when the water is filled. Then you can put your jars in there. Just make sure that the lid is not so tight that this bottle could explode, but not so loose that the lid would fly off. So I sort of screw it maybe 1 or 2 turns. There's our water level, so I'm going to turn that to the next stop, and there'll be water in there to help with the pressure. We'll load in our jars, again, making sure the lids are on just a little bit. Close the chamber. And you can just tighten this to the touch. It doesn't have to be so tight that you can't open it very well. And the knob now should be on the sterilizing mode. I use 134 degrees Celsius, and we'll set that for 45 minutes. It will heat up and pressurize and be done and sterile in 45 minutes. In autoclave for an hour, and then we've put it into a microwave to remelt it down for about 5 minutes. It's still quite hot, so you're going to want to have hot pads. And we're going to pour this agar now into Petri plates. The Petri plates come sterilized. They've been UV sterilized. They don't need to be put into the autoclave. In fact, if you did, they would melt. And we want to make sure, though, that our surface is sterile that we're using. So there's where your Clorox wipes come in handy. You can wipe down the surface where you're going to work. And then you can go ahead and open this sleeve of Petri plates. They usually come in a set of 20. You will be contaminating the outside surface of them just by handling them, but we're not going to get any contaminants on the inside where we're going to grow our bacteria. So I like to take them out in sets of 3 and stack them. The crisping experiment that we're doing is making bacteria cells resistant to an antibiotic. The antibiotic that we're going to be making them resistant to is something called streptomycin. And so in order to see if our experiment has worked, we need to be able to grow the bacteria on a media that has streptomycin in it, and they should be able to live if the crisping experiment has worked. So to add the streptomycin antibiotic to the media, we need to weigh it out. It's a powder, and we'll use our balance again. And remember, you are handling an antibiotic here. It is something that could kill other bacteria in your system, so just handle it with care. And we'll weigh that out, the appropriate amount, and then we will put it into a clean tube. Again, you can use this as a funnel. And add the appropriate amount of distilled water to make the concentration correctly. Once you've done that, it'll be in a solution, and that's where we need to still sterilize it. I know it's an antibiotic and it kills most germs, but it doesn't kill all germs, so it does need to be sterilized in solution. So to do that, we're going to use this filter sterilizer, opening the package and our syringe. And the outside of the syringe won't be sterile, but the liquid we're drawing up will be. So we'll use that and we'll drop the liquid through this 1 micron filter. And now the liquid that I have in the syringe is sterile, and I could place that into another tube to use later. And then you will take your Buxton burner, which we're going to use to sterilize the lip as we open up the cap. And so, a little gas out. Now, cool enough that I can lift it, but not so hot that I'll drop it, or that it'll solidify, and not so cool that it'll solidify on the spot. So take open this lid, and now that I'm taking the lid off, I want to not be talking too much because I don't want to blow bacteria into the bottle. And I'm also going to only keep the lid off as little as possible. So take the lid off. For the next set of plates, I'm going to flame it again on the lip so that any drips coming off won't get contaminated. You want to pour your plates fairly thin and then you can swirl it just ever so slightly to cover the rest of the plate. Notice how I call it clamshelling, just opening the lid enough to pour it in, but not so much that a lot of air is bacteria will drop into the plate. So I'll do that next set of plates there. Now, some of the plates are going to need to have the antibiotic put into the media. So that's where we're going to go back to the sterilized streptomycin that we used. And we're going to add that into the media bottle. You can't actually use an autoclave with the streptomycin or the other antibiotics. It will degrade the antibiotics. So that's why we had to use that filter sterilization method. And now that this is cool enough, it's not going to harm the antibiotic in any way. But we're going to need to add this sterilely to the media and then pour the plates with that in there. So to do that, we'll need to use the micropipettes. The antibiotic solution. Open this just for a quick second. Put that into there. Swirl this out around a little bit. And I do want to label this properly now that I have streptomycin in there. And I also want to label the plates to know that these have the streptomycin or the antibiotic in there. I like to just put a stripe of red Sharpie down the side, and that distinguishes from the other plates that I've poured that are just LB agar alone. And same thing, we will sterilize the lid of that bottle and pour these plates. And just so you know what they look like after they've set, I've pre-made some, and I have them over here. They're no longer liquidy, and the agar has solidified, so you can turn them upside down. And then I use either the bag that they came in or another bag. This is not sterile, and neither is the outside of the plate. But when you store them, it's good to put them upside down with the lids going down, and then you can bag this and put it into the refrigerator. The reason for the lids facing downward is sometimes there'll be a little bit of condensation that will come out of the agar, and you don't want that falling back onto the plate agar itself. It's better to keep that onto the lid. These agar plates that have just set are just the food and agar, and there's no bacteria growing on them yet. So we can store them in the refrigerator, and that will slow down any contaminant growth, and they can store up to several weeks in the refrigerator until we're ready to use them.
Great.
Secondly, you're going to need to grow some bacteria on those plates, and you will purchase a culture of E. coli bacteria, and it will be a strain that is not pathogenic, and the strain will come in a little tube that's called a stab, and you'll remove some of that bacteria from the culture and streak it onto a plate and give it some time to get started growing. We're going to need to grow some bacteria to use in our experiment. So you can order different strains of bacteria. This happens to be E. coli, and don't freak out about E. coli. Some strains can make you quite sick. This particular strain, HME63, is not a pathogenic strain. And when it arrives, it'll be in what's called a stab, a little bit of LB agar where it's been stabbed into that and it's growing. And this can stay in this container for years, and it'll stay in a dormant state. But what we're going to do is grow it and allow it to start to replicate on a plate. So I'm going to start my Bunsen burner so that I can sterilize this inoculation loop. This is an inoculation loop. And need to make sure that that wire is red hot. Open up the container. Notice I'm not setting the lid down on the table. Sometimes I just put the wire on some agar to cool it for a minute, and then I stab it into the area where you can see the bacteria actually growing. And you won't see any bacteria on the loop itself, but there is some on there. It's very small. And then we'll use this and we'll make a little streak, a little line. And then I'm going to sterilize the loop, and I'm going to drag that line across in another direction in order to dilute it slightly. So I'm going to just touch the loop to an area of agar with nothing on it to cool it. You might hear a little sizzle noise. Then I'm going to stripe it across, dragging some of the bacteria from my first stripe. I'm going to turn it a third time and sterilize the loop again, cool it, drag some of the bacteria across, and one last time, rotating the plate, make one more last streak to really dilute those bacteria, and then I'll sterilize that before you're done. And the idea is, in the very first streak, there were tons of bacteria cells. By the time I got into that fourth streak, individual cells will land on one spot on the plate and make a little colony or a little pile that we'll see. So after this will be put into the incubator for 24 hours at 37 degrees Celsius. The plate will look like this, and as you can see where I had those streaks, there's quite a lot of bacteria, and then by the end there's individual colonies of bacteria. We want to have an individual isolated colony of bacteria that's even and alike to start with for our experiment. After you've streaked the bacteria out onto the plate, it's a good idea to write down what you've put onto that plate. So write on the bottom side of the plate, and as small as you can around the outside edge. I like to put my initials, today's date, and the kind of bacteria that's on there. So E. coli strain HM-E63. Then this can go into the incubator, and I put it upside down, believe it or not, in the incubator so that if there's any condensation, it'll fall onto the lid, not fall back onto the bacteria itself as it's trying to grow. I'm putting them with the lid down in case there's any condensation that can fall onto the lid. Close the incubator, and we'll come back and look in on it in 24 hours. We're back after 24 hours, and we're going to check to see that we have bacterial growth. So this is the first step in our experiment. We do have good bacteria here. That's what you want to check. If you don't have growth on the plate, it may be because the original stab has gone bad. You may not have picked bacteria onto the loop. You may have burned the loop a little bit too much and didn't get bacteria onto the plate. So this is a key step to check. Congratulations if you got bacteria and you're ready to move on to doing the CRISPR experiment. Next, you'll need to make competent cells. And remember, the cells are the bacteria, and what competent means is able to uptake DNA from the environment. So here I have a picture of an E. coli cell. It is bacteria, it is a cell. It happens to be of the type of bacteria E. coli. And again, it's not a dangerous strain of E. coli. And we are going to have DNA from the environment that we want the cell to take in. The problem is that the wall of the cell is negatively charged, and DNA also is negatively charged due to the phosphate groups in DNA. So there's a repulsion that would happen between the DNA and the cells themselves. So we're going to add a solution of calcium chloride, which will provide calcium ions that will neutralize those charges and allow the DNA to settle on the surface of the bacteria cells. So now we're really going to get started with crisping and start our experiment. The first step is using sterile microtubes and putting the bacteria into the tubes. Remember, this is sterilized, so the outsides are not sterilized, but the inside is. And before we put the bacteria into the tube, we need it to be in a liquid. So that's where our calcium chloride comes in. And I'm going to label one of these tubes with a plus. That will be our CRISPR experiment, and one with a negative that we're going to have for a negative control. And then we're going to add 100 microliters of the calcium chloride to each of those tubes. So make sure you have the correct micropipette and the correct size of tips. Open that. Take out the calcium chloride. And calcium chloride is used to neutralize the charge of the bacteria cell wall, as well as the negative charge on DNA, because we need to get the DNA into bacteria cells, and since DNA is negatively charged and cell walls are also negatively charged, they will typically repel each other. So the calcium positive 2 ions will neutralize that charge and allow the bacteria to come close to the surface, so for the DNA to come close to the surface of the bacteria. Now that I'm done with the calcium chloride, I'm going to deposit that tip there. So what I've just done is added a 25 millimolar solution of calcium chloride into 2 tubes. One is our negative control, and one will be the tube that we're going to do the CRISPR experiment in. Now we have to add bacteria to the solution of calcium chloride. So in order to do that, I'm going to pick a colony off this plate sterilely. I'll start my Bunsen burner again. Use my inoculating loop. Sterilize that. And I'll cool it just a tad on the side and pick off one individual colony of bacteria. You may not see it on the loop. Turn the Bunsen burner off. And I'm going to take the lid off of this, being careful not to touch the inside surface of it. And I will dangle this loop into the liquid, and cells, believe it or not, are coming off into the liquid right now. And then I will cap that again. And then we'll do the same with the other tube for our negative control. I'm going to do a second tube with bacteria in it that's not going to get crisped, so that we will know that it indeed is not antibiotic resistant. So we'll need to start the Bunsen burner one more time. Pick off another colony, sterile loop. I'm not going to open this plate very far. Take one single colony off the plate where I see one dot. And bacteria will go off into that liquid. I'll close that. And those will get everything except for the DNA that we're going to add to the crisping tube. Next, we will be adding our DNA to these competent cells. So we've added the calcium chloride, then we will add the DNA, and we'll wait a little bit of time so that those charges get neutralized, and the DNA will start to settle on the surface of the bacteria. I'm going to add DNA to my tube, which has the bacteria in there, which will transform the bacteria, allow them to make the Cas9 enzyme, to have the guide RNA, to find the region that it will dismantle, and to the template DNA that we're going to use to edit our genome. So we will put 10 microliters of each type of DNA into that tube. So set the pipette to 10 microliters, which is a very small volume. And take out your first DNA, which is a plasmid that has the Cas9 enzyme gene. And pipette, 10 microliters. The DNA is kept in the freezer, and so right now I have it in a cup of ice. Then I will add that to my solution. And get a new tip for my second kind of DNA. The next DNA is the guide RNA, and that will bind to the Cas9 enzyme and to the bacterial genome DNA and make a cut at a specific location. So again, 10 microliters. And we can't use that pipette again. Dispose of that. And lastly, the third type of DNA is the template DNA. We're going to flood the system with enough template DNA to trick the cells into incorporating this DNA into the location that got cut, and we'll have edited the genome. The negative control is not going to get DNA, but we need to make sure that it has the same volume so that the concentrations are the same of the cells. So we'll set our micropipette to 30 microliters, since we've just added 3 times 10 microliters. So we set that. And use sterile water to replace the same amount of DNA. So remember, we have this water sterilized in autoclave. I'll put that into the tube that I've labeled with a negative. These tubes need to sit on ice for half an hour, so I'll put those into my little cup of ice. And what's happening now during that time frame is that the DNA is settling on the surface of the bacteria cells, and the calcium chloride is neutralizing the charge so that the DNA will come close to the cells. At this point, the DNA hasn't entered the cells, but we're just letting it settle. The fifth step is to do a heat shock. So you have a tube right now that has bacteria cells, it has some solution of calcium chloride in there, it has the DNA solution in there, and at this point, hopefully the DNA is settled on the surface of the cells. But we want the DNA to get into the cells, so those tubes are going to be starting out on ice, and we will transfer them for a quick 30 seconds to a 42-degree water bath. And that will allow the pores of the bacteria cell wall to open, and the DNA will actually diffuse in. It'll be more concentrated on the outside than on the inside, and the natural tendency of molecules is to move to a lower concentrated area. And so that little bit of heat will open up the pores, and then we'll put it back on ice to close up the pores, and we've hopefully trapped inside of the cells the DNA that we want to introduce. So after the half an hour on ice, we're going to do a heat shock where we put these tubes for 30 seconds into a water bath that's been set at 42 degrees Celsius. So we'll put those into our little rack here, and we're going to time for exactly 30 seconds. And after the 30 seconds, you will take those tubes out and put them right back onto ice so they go from a heated tube to cool down quite quickly. During the heating process, even though it's a short time period, the cell membranes are opening up their pores and allowing for the DNA to diffuse inside of the cells. And then the cell membranes will close up again once you put them back on ice. So it's just a simple step of a heat shock that allows the DNA to get into the cells. If these were mammalian cells that we were growing in culture, we would need to use a different method. Fortunately, mammalian cells, it's a little bit harder to get the DNA inside, especially because they have a nucleus, so the DNA needs to go all the way through 2 membranes. So usually electroporation is used, a little electric shock to cause the DNA to be uptaken by the cells. The next step is to feed the bacteria. We'll give them a little bit of LB broth, which is a food source, and it'll help the bacteria to be able to multiply and also for this CRISPR mechanism to be taking place. So take your sterile LB broth, pipette up 500 microliters, and adding that to this tube. Tips, LB broth can be easily contaminated. And that, the other tube. And now these tubes can either be put in the incubator at 30 degrees for 2 hours or left out on the desk for 3 hours at room temperature, 3 to 4 hours at room temperature. And the crisping is taking place right now as we speak. Then we will transfer the cells onto agar plates, to transfer the bacteria onto agar plates and spread it, and then we will incubate for 24 hours at 30 degrees Celsius. And in that time period, the cells will be growing on the plate, and then we'll come back and look at the results and see if we've got bacteria that have been transformed. After waiting the several hours for the bacteria to recuperate, we need to spread them onto plates, and there they will grow and continue to have the CRISPR reaction taking place, and the plates will also have the antibiotic on them to eliminate any bacteria that did not get crisped. So in order to move them from these tubes onto the plates, we need to do that in a sterile way. So we're going to use some rubbing alcohol or ethanol, 91%, and a glass spreader. And again, I will need the Buxton burner. And I'm going to move 200 microliters of this solution onto the plate with micropipette. So I'll pipette that up. Put that onto the plate. And I'll put 200 microliters onto a plate that's just LB and agar, and another 200 microliters onto a plate that had the streptomycin antibiotic on it. Then take this glass rod and I'll sterilize it in the flame. Don't hold it in the flame too long or the glass will start to melt. And then I'm just going to spread this onto the plate, almost like making a crepe, and just make sure that the liquid gets evenly distributed across the plate. And I'll even sterilize it in between plates, even though that was both the same, coming from the same original bacteria. Spread that around. Sterilize that. Put that back in. And then we want to label the plates so we know what we've done. This plate is the E. coli strain HME65. I'm going to put my initials and the date. And this is the plate that has the CRISPR added to the E. coli, so we're going to put plus CRISPR. The other plate is the same except for it has streptomycin in addition to the LB agar. So I'll write LB agar plus strep, and then E. coli plus CRISPR, and my initials and the date. This plate I should write that it's LB agar only. And then these plates are going to go just like they are now, upside down, stacked in the incubator for 24 hours, and then we'll be able to see our result. We also need to prepare plates for our negative control to be sure that we had bacteria to start with and that it did not get crisped for our negative control. So we'll write on these plates first that it's just the E. coli strain and no CRISPR. My initials and the date. And we'll move, and then again, this plate with the red stripe has the streptomycin on it. We will move from the negative control to, again, another 200 microliters of our mixture. I'm going to use the same tip this time. And I'll spread and use the glass spreading rod. Cool it a little bit on the side, spread it around. Spread that all around. All four of these plates can get stacked just like that and put into the incubator. And then we will be able to come back and look at our results and see if we got bacteria that have been transformed. The first plate is the plate that had the kanamycin in the media. It is the E. coli that had the CRISPR DNA added. And as you can see, there are colonies of bacteria growing on there that are resistant to the streptomycin antibiotic. And that's what we want to see. So we could even count the number of colonies that were successfully transformed and crisped. Remember, we had also used a negative control where we didn't give it any of the CRISPR DNA, and there's nothing growing on this plate that also had the streptomycin on it. So it does show that even though we spread bacteria on there, it couldn't grow because it's not resistant to streptomycin unless it's been transformed to do so. We also had two other plates where we grew bacteria on LB, plain old LB agar, and the reason for that was just to check that we had bacteria to start with. So especially on the negative control, we just spread E. coli onto an LB plate. Normally it should grow on that, and indeed it did. It grew so much that it created a lawn. So it's actually everywhere with a slight milky look to the plate, different from this one. So we know we had bacteria to start with, but it didn't get transformed. And then same with the crisped bacteria, it is growing in a lawn on the LB as you would expect. But since there's no streptomycin on there, we can't tell if the experiment has worked or not. So that's why this is our key plate here. So yay, we've been successful with this experiment.